Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome everybody again to another episode of our show, bringing you another really fascinating guest today, helping to create a better tomorrow for many people. Today we have the honor of being joined by Dr. Thomas Johnson, who is a glaucoma specialist and the Alan and Shelley Holt Rising Professor in Ophthalmology at the Wilmer Eye Institute, Johns Hopkins University. Dr. Johnson received his bachelor's degree in biologic sciences from Northwestern University. He was a Gates, Cambridge, and NIH-OxCam Scholar, earning his PhD in Clinical Neuroscience from the University of Cambridge. And then, after all that, he completed his medical training at Johns Hopkins School of Medicine and served as an intern on the Johns Hopkins Osler Medical Service prior to completing his ophthalmology residency and glaucoma fellowship at the Wilmer Eye Institute. Dr. Johnson's research interests are quite broad and focused on understanding the pathophysiology of both retinal and optic nerve neurodegenerative disorders, and on the development of both neuroprotective and neuroregenerative therapies for these conditions. His thesis work evaluated intraocular stem and progenitor cell transplantation as one of the possible neuroprotective therapies for glaucoma, and his research contributions have been recognized all over the place with the World Glaucoma Association Award nomination, the National Eye Institute Scientific Directors Award, and the Association for Research in Vision and Ophthalmology's Merck Innovation Ophthalmology Research Award. Dr. Johnson also founded and served as the director of Student Sight Savers Program, a program that helps provide vision screening services to low-income residents of Baltimore, helping them to obtain access to clinical ophthalmological care. And presently, he is interested in the range of neurobiologic processes that ultimately lead to retinal ganglion cell death and dysfunction in glaucoma and a range of other optic neuropathies, in particular seeking to understand the molecular mechanisms underlying axonal degeneration, dendrite retraction, and afferent synapse loss and cell body death in glaucoma, and looking at various ways to ultimately create therapies to reverse these processes in this area of significant unmet medical need. All that being said, we're honored to have him with us today. Dr. Thomas Johnson, thank you so much for taking the time out of your schedule to talk to us for a bit.
No, of course. Thank you very much, Ira, for having me. I'm a big fan of your podcast, and so it's really a special opportunity to get to speak with you today. Thank you.
Yeah, no, it's really great. I've been looking forward to this discussion for a while, and you know, I would love to, before we jump into the details of everything you're doing, I would like to, as we typically do, sort of give you the mic for a couple of minutes just to talk a little bit more about yourself, introduce yourself to the audience. If you could take us back a little bit, introduce us to sort of, you know, who you are, where you grew up, how you got interested in not just biology, but medicine, neurosciences, a little bit of those early days, so we can get to know you a little better. That'd be great.
Sure. Yeah. Well, you did a fantastic job rehashing my training and my experience so far, so I appreciate that. You know, I think my interest in medicine and neuroscience started when I was a kid. I grew up outside Chicago, and my dad is a veterinarian. And so when I was young, even from the age of 6 or 7, I would spend Saturdays in his veterinary clinic helping out with processing lab samples and checking in his patients, which were dogs and cats. And I was just really fascinated by medicine, the ability of medicine to heal sick patients, and, you know, see dogs and cats and other small animals come in and really turn around from being very ill, very unhappy, not feeling well, to seeing what medicine could do to bring health back to living things. And so by the time I hit high school, I became more interested in human medicine, and did a number of shadowing opportunities, local high schools and things like that. And so I knew I wanted to be a doctor from the time I was in high school. And then I went to Northwestern for undergraduate and really fell in love with neuroscience, seeing how relatively what seemed like simple cells that can convey electrical information can coordinate themselves into circuits and organize themselves into tissues like the retina and the brain, and eventually lead to things like vision and conscious perception. It's just an amazing thing how the body is able to create sort of a supercomputer from scratch that is able to perceive the world around us. It's really amazing. So I think a lot of my training and my early experience is benefited from amazing mentorship. And so I think that's an important take-home message that I want to relay is in medicine and in science in general, the mentors that lead you through your professional development and training are really the people that open up doors to possibilities for what you're going to do for the rest of your career. So I've benefited from tremendous mentors throughout the course of my training. When I was at Northwestern, I had a good friend named Lucinda Camrus, whose father was Carl Camrus. He is an ophthalmologist. He was chairman at the University of Nebraska Medical Center, and he's famous in the ophthalmology world for having discovered prostaglandin analogs, which are the type of eye drop that's now the #1 therapy to treat glaucoma. He did this when he was an undergraduate at Yale, made the discovery that if you formulate these prostaglandins into eye drops, it lowers the eye pressure. So given my interest in medicine and visual neuroscience, I spent a summer and then later a year with Carl at the University of Nebraska. And that's where I really fell in love with ophthalmology. I got to shadow him in the clinic, shadow him in the operating room when he performed surgeries on patients' eyes, and really decided that, vision and ophthalmology meld my passions of helping people from a medical perspective, help them with their health, and then also provides a really rich area for studying neuroscience and how neurons contribute to visual perception. From then it just was a series of experiences and training with various mentors that have really gotten me to where I'm interested today. So when I was at Nebraska, I met Yoni Kitness, who is a PhD neuroscientist and is now chair of neuroscience at WashU. He is now famous for having discovered the fact that there are lymphatic drainage pathways in the brain, something that was thought to be not true for decades and decades. And he's kind of turn the field over on that. So he got me interested. He gave me my first experience in basic science, studying cells in culture and animal models of neurodegenerative disease. And it was based on that experience that I decided to defer my plans to go to medical school and actually pursue a PhD in neuroscience. And so, as you mentioned, I did that half at the University of Cambridge and half at the National Eye Institute in Bethesda, and had a couple of fantastic mentors there. Keith Martin is a glaucoma specialist who was my PhD supervisor and is now in charge of the Center for Eye Research Australia, which is a massive ophthalmology and visual science enterprise. And I worked with Slava Tomarev at the National Eye Institute doing molecular biology. And so after completing my PhD, I came back to my original plan, which was to earn an MD, become a physician so that I could treat patients in the clinic and the OR. And so I've been at Johns Hopkins ever since, working again with amazing mentors. Harry Quigley, who is a world-renowned glaucoma specialist and made many of the seminal discoveries that help us understand how and why the optic nerve degenerates in glaucoma, has been a mentor of mine. And then I also work closely with Don Zach, who is a world-renowned stem cell molecular biologist who's really taught me a lot about what we're doing now with stem cells, differentiating them into retinal ganglion cells, and trying to figure out how we might be able to get those to replace dead optic nerve neurons to restore vision.
Excellent. Excellent. I appreciate that that additional background, because it really is a fascinating journey that you've been on, not just in the research domain, but in the clinical care side of things. So it's a really exciting intersection of it all. When one looks at, I've worn glasses when I'm on right now, but I've worn glasses since I was 9 years old. I would go to the ophthalmologist, I get the little glaucoma test, never think much of it. But then, diving into the some of this data from recent days, around 3 million Americans alone living with glaucoma, 2.7 million or so over the age of 40. We have about 80 million worldwide, and some of the projections to the middle of the century, upwards of around 115 million or so. Needless to say, a major unmet medical need. We talked a little bit about age-related macular degeneration on the show, and there seems an area that sort of, some of the biologics and so forth have sort of laid some groundwork in terms of disease modification and so forth. With glaucoma, as you were mentioning, a lot of the pharmacotherapeutic space has primarily existed in how we reduce pressure, how we reduce inflammation, and some of that fluid flow. Talk about, if you would, a little bit about where we are in terms of the glaucoma pharmacotherapeutic space overall, and we'll get into, obviously, your work in terms of neuroprotection, neuroregeneration, and so forth. But a little bit of sort of why glaucoma has, where it's been sort of in the scheme of things for pharmacotherapeutic development over the years, because we made progress, but we still have a little way to go.
Yeah, absolutely. So glaucoma is a fascinating disease, and it is a major medical and public health problem. And part of the reason for that is that in most people, the early stages of glaucoma are completely asymptomatic. It has this colloquial name, the sneak thief of sight. And that's because glaucoma typically doesn't cause pain. And as it begins to take away vision, it does so very slowly. And it starts in the periphery, way out in your side vision, and can take away a tremendous amount of your peripheral visual field before it begins to affect your central vision. And so it happens so insidiously that many people don't recognize that they have this disease until very late stages. And so one of the key aspects of glaucoma is identifying it. And for most people, it's only going to be identified at an early stage if you happen to see the eye doctor and they not only check your eye pressure, but also take a look at the appearance of the optic nerve in the eyeball. And that's important because eye pressure is an important aspect of the disease, but it's definitely not the whole story.
Sure.
We used to think that if you had an eye pressure above this magical number of 21 millimeters of mercury, that means you have glaucoma. And if you're less than 21 millimeters of mercury, then you're safe. But epidemiological research in the last few decades has demonstrated that there are lots of people out there all over the world walking around with pressures well into the 20s that live like that for decades and never develop glaucoma. Mm-hmm. And half of people with glaucoma actually have never had a measured pressure above 21 millimeters of mercury. So we now understand that glaucoma is related to the intraocular pressure. And if you lower the intraocular pressure in a patient with glaucoma, their disease will slow down or stop getting worse. Mm-hmm. But there's a lot that has to do with individual susceptibility to various levels of pressure. Some people have very resilient optic nerves that can sustain high intraocular pressure, and they don't develop optic nerve degeneration. So those people don't have glaucoma, mm-hmm. They just have high eye pressure. And other people have what would be a normal intraocular pressure for most people, but for whatever reason, they develop progressive loss of retinal ganglion cell axons in the optic nerve, progressive optic nerve degeneration, and progressive vision loss. And a lot of research is trying to determine what is special about those people. Why are their eyes so susceptible? There are theories that it has to do with the biomechanics of the optic nerve head in the back of the eye. It's interesting. The optic nerve, we should just talk about briefly, is actually, it's a structure that's sort of like a fiber optic cable. And in that cable, it has hundreds of thousands or millions of retinal ganglion cell axons, which are the fibers of the neurons that carry visual information from the eye to the brain. And the retinal ganglion cells have cell bodies that live in the retina, which lines the back wall of the eye. And they extend this really long axonal fiber that goes through the retina. Then when it gets to the optic nerve head, it goes out the back of the eye and into the brain. So right at the optic nerve head, that retinal ganglion cell axon is actually making a 90-degree turn in space to get into the optic nerve. And so there's thoughts that as the eye pressure elevates, there's something about that structure where that 90-degree turn is made that somehow kinks or damages structurally the axon itself. And there's been studies that show there's normally transport of proteins and other materials through the optic nerve going in both directions. And if one were to experimentally raise the eye pressure in a model of glaucoma, you end up with a blockade of that transport, which probably initiates an injury signal to the retinal ganglion cells. So some people just may have a structural defect in their optic nerve head that leads to that injury signal being generated at relatively lower pressures. And then there's also thought that blood flow is important in the optic nerve. And as you raise the pressure in the eye, blood has a harder time getting into the eye and perfusing, delivering oxygen and nutrients to the optic nerve axons. So all of that is a very interesting and lively area of research in glaucoma. But at the end of the day, what we know is that we can slow or halt disease progression if we lower the intraocular pressure of a patient with glaucoma. And so at this point in time, all of the treatments that we have for glaucoma are focused on that one thing, lowering intraocular pressure. We have three main ways of doing that. Number one is with therapies like eye drops. There are a number of different types of eye drops that all lower the eye pressure in different ways. Some of them slow down how fast the eye is producing internal fluid called aqueous humor to keep itself pumped up. Some of them increase how fast fluid is drained out of the eye through a number of different pathways. There's also laser procedures that can be done in the clinic that can enhance the outflow of fluid from the eye and lower the pressure. And then as an ophthalmologist, I perform a number of surgeries that actually help shunt aqueous fluid out of the eye, either through manipulation of the eye's normal drainage pathways or through implantations of things like shunts and tubes that actually drain fluid out of the eye. So as far as getting back to your original question, the drugs that we use, the medications that we use to treat glaucoma now, there are a number of different classes of medications, and this is an area of active research. In fact, we just had 2 new FDA-approved medications added to our repertoire about 3 years ago. One is a Rho kinase inhibitor that increases aqueous outflow through the trabecular meshwork and probably reduces aqueous production a little bit as well. And the other one is a nitric oxide donor that is added to an existing prostaglandin analog that helps reduce eye pressure by increasing outflow as well. I think the number one area of unmet need, though, is in trying to move away from the paradigm where patients need to take eye drops multiple times a day. And that is because adherence to glaucoma medications is a major problem and really responsible for continued loss of vision in a huge number of glaucoma patients. And the reason for this is multifactorial. You know, I think getting back to the fact that glaucoma is a generally asymptomatic disease, it's often hard to remember to take medicine for something that is not causing pain or causing active symptoms. Your glaucoma for most people is not at the forefront of their mind all the time. So just like it can be tricky to remember your cholesterol medicine or your blood pressure medicine, it can be tough to remember to put in your glaucoma eye drops. Some eye drops are just once a day. Some of them are 2 and 3 times a day. And so glaucoma eye drops only work to lower the pressure when you remember to put them in. And people that have studied the adherence of patients with glaucoma to their medicines have uncovered the fact that glaucoma eye drops are taken far less frequently than they actually should. There have been studies where people have put electronic eye drop monitors on the bottles so that they record when the patient actually inverts the bottle and puts it into their eye. And something like 75% of patients don't actually take about 75% of their medicines. So, you know, only 25% of the patients are taking the eye drops as frequently as they should. And that's in studies when they know they're being monitored because there's a monitor on the bottle. So in real life, there's probably even less medications actually making it into the eye than there should be. So how can we get around that? Well, there's a big push now for sustained delivery of glaucoma medications through various formulations. We have some investigators here at Wilmer that are studying nanoparticle type delivery devices that can be put into eye drops that only need to be taken maybe once a week, and then work over long periods of time by developing a film on the surface of the eye that can release drug for several days at a time. There have been some attempts to make implantable devices that go in the tear ducts in the eyelid and can secrete glaucoma eye drops onto the surface of the eye. There's also been some work in developing either contact lenses or implantable plastic rings that fit underneath the eyelid. And it sounds uncomfortable, but once they're in, patients kind of forget that they have them. None of those things are available yet, but I really think that if any of those make it to market, they'll really transform the landscape of medical treatment of glaucoma. Because we already have a number of molecules that we know lower the pressure and reduce the risk of vision loss. And we just need to find ways, better ways to get them into the eye. We do have one new medicine that is an injectable form of a prostaglandin analog. It requires the doctor to actually inject it into the front part of the eye with a needle. But once that's done, it then secretes and provides this drug over the course of months for the patient without them having to remember to take eye drops on a daily basis. So that and other forms of potentially injectable drugs, I think would really benefit the glaucoma community.
Yeah, the issue of compliance, obviously, you know, is an issue across sort of the pharmaceutical spectrum when it comes to, as you were mentioning, the eye and getting things in multiple times a day, and then obviously it hurts and and all that. That I understand. It's as a pharmacist, right. For 30 some odd years, I completely understand the potential, not just for these new delivery vehicles, but in some cases, sort of as you were saying, some of the repurposing of molecules and so forth. So that's very exciting. But so yeah, I mean, Tom, thank you for obviously that overview of where we are now going into sort of the next generation and the cutting edge stuff, which you're actively involved in. And I look at these, and I know there's overlap here, but I sort of look at these two silos, one in terms of neuroprotection, and then the other neuroregeneration. We can start with neuroprotection first, and you've published extensively over the years looking at stem cells in neuroprotection, stem cell derived secretome materials in neuroprotection, and then there's even I love going into our guest publication record, but there's this really cool paper I saw from 2007 where you were looking at protective autoimmunity, using using bacterial DNA. Really, really neat stuff. When it comes to some of these neuroprotection strategies, though, what are some, because obviously there's so much going on in there in terms of what happens in between some damage event or pressure event and blindness, inflammation, fibrosis, immune response of different types. What are some of the mechanisms that you are looking to impact in terms of these neuroprotection strategies, whether you're using the stem cells or the secreted entities or other biologic factors endeavoring to slow down degeneration or increase neuroprotection?
Yeah, there's a huge need for neuroprotective agents in glaucoma. And I think this goes back to some of the things we've already talked about.
Yeah.
There are patients where we can lower their pressure down to very low levels, 12 millimeters of mercury, and yet they still continue to have progressive ongoing degeneration of their optic nerve and loss of vision. And in patients like that, because our treatments for glaucoma are at present only related to lowering the intraocular pressure, there's sometimes little additional treatment that we can offer to patients. You can surgically lower the pressure lower to 8 or 7 millimeters of mercury, but at some point you hit a very narrow therapeutic window, because lowering the pressure below a certain threshold can cause dysfunction to the eye, blurred vision, and other problems. So that's one group of patients that would benefit from neuroprotection, is people that have already achieved very low pressures, but have such susceptible nerves or eyes that they're continuing to lose vision. And then there's the group of people that just, for whatever reason, adherence or side effects or other issues, can't take the eye drops, or the eye drops aren't working as well as we would like. And, despite our best efforts as glaucoma doctors and surgeons, we're just not able to lower the eye pressure as much as we would like. In those two groups of people, it would be great to have a second therapeutic target, some kind of a mechanism where we could protect retinal ganglion cells from dying that was independent of intraocular pressure. And that's essentially what neuroprotection is. It's looking for alternative targets that don't directly address eye pressure. This is a field that's been busting for 20 or 30 years now, and there are hundreds and hundreds of papers of molecular pathways in retinal ganglion cells and in the other related cell types that accompany them in the retina and the optic nerve, like astrocytes and Mueller glia and other types of support cells. In a number of experimental models, like cell cultures or animal models of optic neuropathy, investigators have been pretty successful in targeting various molecular pathways to improve retinal ganglion cell survival and reduce disease states. But somewhat disappointingly, none so far have made it past clinical trial. I think there's a number of reasons for that. One is that some of our preclinical scientific models don't adequately model human disease. So what works in a mouse or a rat doesn't always work in a human being. And there's also just been logistical hurdles in getting potential targets through the regulatory process. Mm-hmm. So we were very close to having our first neuroprotective agent for glaucoma about 10 years ago. In a drug called memantine that was trialed. It's actually approved for the treatment of Alzheimer's disease. Yep. And there was some preclinical data in animal models suggesting that it might work in glaucoma as well. And that was tried in a large human clinical trial, and unfortunately it failed. That trial cost hundreds of millions of dollars to conduct. And so unfortunately, I kind of soured the field and pharmaceutical companies and things from pursuing this for some time. Although I'm happy to say that I think we're now emerging from that into an area of high interest again in neuroprotective treatments. And I'm aware of several companies that are currently conducting or planning human clinical trials for a number of neuroprotective agents in glaucoma. So it may be just a few years before we have our first treatments available. As far as targets, I think there are a number of different pathways that are of great interest. One that we've been studying here at Johns Hopkins is called the dual leucine zipper kinase molecule, or DLK. And this is a particular intracellular kinase. It's a protein that phosphorylates other proteins.
Yep.
And it is critical to linking the damage signal that occurs at the axon when high pressure causes damage to the axon, to sending the message back to the retinal ganglion cell that it's been injured, and so it should start the apoptosis process, where it's going to undergo regulated cell death. And if you block or remove the DLK message, then the cell actually survives a number of different insults, not just glaucoma, but in experimental paradigms, toxins and mechanical trauma to the nerve, and a number of things that may prove neuroprotective. So I think that's one molecular pathway that we've been interested in. There's also been a lot of great work coming out of Pete Williams's lab that started in animal models and is now in human clinical trials. And that relates to oxidative metabolism and nicotinamide, or vitamin B3.
Okay.
It has been shown in animal models to be impaired when glaucoma occurs, and supplementing nicotinamide at very high levels is robustly neuroprotective in models of glaucoma. So that's now being tested in human subjects. And there's been a couple of papers published in the last 2 years that demonstrate when human patients with glaucoma take nicotinamide, it increases their visual function. Now, these were relatively short-term studies. There's a lot of caveats here, but in tests of their electrophysiologic function of the nerve and communication to the brain, and in tests of their visual fields, over the course of just 3 to 4 months, there's a signal emerging from the data that suggests the retinal ganglion cells are functioning a little bit better in the group that received this drug. So obviously, I think what needs to be done now are longer term studies in large numbers of patients to determine whether this is actually able to alter the trajectory of vision loss in glaucoma, independent of eye pressure measurements. But if I had to guess, if those studies pan out, that may be our first true bona fide neuroprotective treatment for glaucoma.
Outstanding. Outstanding. And Tom, moving from protection now to sort of the next holy grail stage of this, neuroregeneration. Again, you've been actively publishing here, and as you were just saying, it's one thing. Yeah, we have stem cells. We've gotten pretty good at that. We've gotten good at differentiating them, but then there's the whole world of how one gets proper engraftment, how one places these biomaterials in terms of the retinal ganglion cells and so forth in the right places so that they are functional and they don't just secrete something and then die or go away. You've written on not just the research, but also the challenges. Talk a little bit, if you would, about some of these challenges, because I think we're all sort of, looking at the stem cell space, looking at regenerative medicine in general, we realize this is a holy grail, whether it's 2022 or 2042 or whenever it's all going to happen. But we got to get through some of these other impairments, let's say, and one of the ones that you've written about, you wrote a recent paper about the role of the internal limiting membrane in preventing proper engraftment of these cells. Talk about a little bit of the challenges that you see, and also your work in neuroregeneration, if you would.
Sure. Well, again, you describe it as a holy grail, and I think that's absolutely true. As a glaucoma specialist, this hits home for me because I'm in clinic every week, and without fail, at least once or twice a month, I have a patient come into my clinic who has advanced, basically end-stage glaucoma. And most of the people weren't diagnosed until very late in the disease for the reasons we talked about before, and they have either extremely poor vision or, in some cases, no vision at all. They have a flashlight being shown in their eyes in a dark room. They can't tell if that flashlight is on or off. They have no light perception. And unfortunately, at this point in time, we have nothing to offer those people as far as methods to improve or restore their vision. In order to do that, we would need to regenerate the optic nerve and replace those retinal ganglion cells, which communicate vision to the eye, from the eye to the brain. So, I like to say that this was something that I think people considered science fiction just 20 years ago.
Yep.
The retinal ganglion cells are incredibly complicated. We talk about them like it's one cell type, but there's actually many, many different cell types. In mice, there's more than 40 subclasses of retinal ganglion cells. In humans and primates, the data suggests there's probably fewer than that, but they're not all the same. Some are important for detecting motion, some detect edges, some detect color. So it's not even a case of figuring out how to replace one cell type in the retina and the optic nerve, but really a number of different cell types. And, some people have equated the problem to one of those old school phone boards that you'd have like in the basement of a big office building. Yep. Where you're trying to connect different phones that people call in, and you have to connect the cords to the right places. That's sort of like the problem that we have here. When people are born, they have somewhere between 300,000 and 3 million retinal ganglion cells. And those ganglion cells are positioned within the retina. Yep. To detect light that hits the retina in a precise place. So that place in the retina corresponds to the part of their visual field that retinal ganglion cell is sensing. So retinal ganglion cells that live, for instance, in the bottom of the retina are actually detecting light from the top of your visual field and vice versa. And in order for your brain to make sense of those millions of different signals that are coming in through the optic nerve, those retinal ganglion cells have to connect in the brain in an orderly fashion. They can't just kind of randomly connect to any center. So there's a very precise pattern of how retinal ganglion cell axons connect to the neurons that live inside the brain. So we have written, as you said, about the various challenges that exist to actually regenerating retinal ganglion cells. And the amazing thing is that in the past 20 years, we've come so far in our scientific knowledge and technology that the individual steps for achieving this have probably all been achieved in isolation in specific paradigms. So we know how to take human stem cells and differentiate them into retinal ganglion cells. And we have multiple different ways of doing this. We have directed differentiation cocktails that we can just add different signals to the culture media. And over the course of several weeks, they eventually differentiate into retinal ganglion cells. There's also this really interesting technique of retinal organoids, where you can get stem cells, and it's not just retina, there are all sorts of different organoids where they will self-assemble into a complex organ. There are brain organoids, and retinal organoids will actually begin to form a ball. And then, just like occurs during development, they will invaginate into a layered structure that resembles an eye with a retina, and they will develop the pattern structure of all the different laminated retinal cell layers that exist in the human eye. And so we can get retinal ganglion cells from retinal organoids as well. We are working on figuring out how to transplant retinal ganglion cells into the eye and get them to survive over long periods of time. And we're working on how to get them to grow dendrites into the retina. The dendrites are processes from neurons that receive signals from other neurons. So if you think of the retinal ganglion cell as a two-way communication station, it has ears that listen to the information coming into the cell. It does some processing, and then it communicates the information out the axon. And so the dendrites need to absorb visual information from other retinal neurons, the bipolar cells and the amacrine cells within the retina. So we are figuring out how to get retinal ganglion cell dendrites to go into the correct layer of the retina called the inner plexiform layer, where those communication stations or synapses might be formed. And then we need to get the axon to grow through the optic nerve and into the brain. Now, there's been a tremendous amount of work in the last 15 years or so looking at the regeneration of axons, not from transplanted ganglion cells, but from endogenous retinal ganglion cells following injury. So if you were to take a mouse, for instance, and perform an optic nerve crush with a pair of forceps, all the retinal ganglion cells on the brain side of that optic nerve will degenerate. And a lot of the retinal ganglion cells will die. But if you provide the retinal ganglion cells the right support factors and molecular signals, you can get a large number of them to survive. And you can actually get some of them to regenerate axons and start growing back through the optic nerve. And in some experimental models, they actually make it all the way back to the brain, to those communication stations in the subcortical visual targets that are necessary to support vision. So, at least in that paradigm, it suggests that axonal regeneration to the brain is possible. I think what's necessary now is combining the seminal findings from all of these sort of disparate, isolated experiments and finding a unified protocol that's able to make this happen for transplanted retinal ganglion cells into the eye. And that's what our work in collaboration with a number of investigators all over the world is all about. So right now we're focusing primarily on the intraocular portion of this connectivity, where we're trying to get retinal ganglion cells to migrate into the retina and then create synapses with the cells that are inputting visual information into the optic nerve. And as you alluded to, we started doing this work about 3 years ago by just injecting stem cell derived retinal ganglion cells into the eyes of animal models with glaucoma. And we found that those cells would survive in the eye and they would sit on the surface of the retina and they would grow processes in 2 dimensions on the surface of the retina. But they really would not grow any dendrites down into the retinal tissue. It was almost as if the retinal ganglion cells that we transplanted couldn't see the retinal tissue underneath. That retinal tissue is sort of their native home. We thought that's where they would like to be, but they behaved as if they were just growing on tissue culture plastic. And so we did a variety of experiments to try to figure out what is the barrier, what's the signal that's preventing them from actually integrating into the retina that was directly below them, a matter of micrometers away from where they landed on the retina. And there's a structure on the retina called the internal limiting membrane, or the ILM. This structure is only a few micrometers thick. It's a basement membrane composed of things like laminin and collagen 4 and perlecan. And it's really important for retinal development. If you don't have an internal limiting membrane, there are defects in how the retinal ganglion cells pattern themselves. They don't form a single layer. They form these clumps. Some of them migrate out of the retina. There's also defects in how the glial cells pattern within the retina. And because the vasculature in development follows a glial scaffold, you get blood vessels that grow out of the retina and into the vitreous. And you end up with these sort of bizarre and very atypical retinas. But that's only during development. Interestingly, once the retina is developed and someone is an adult, the ILM appears to be largely dispensable. And we know that because the ILM is actually involved in a set of pathological states for the retina that are related to the atypical adhesion of the vitreous gel to the ILM, and sometimes to a little membrane that can grow on top of the ILM surface called an epiretinal membrane. And sometimes when the vitreous starts tugging on the ILM, it can distort the surface of the retina, make the retina wrinkly, which can cause your vision then to become wrinkly or distorted. We call that metamorphopsia. And sometimes the vitreous can pull on the retina so much that it actually causes a break to form right in the center of the vision. We call that a macular hole. The treatment for a macular hole is actually to surgically remove the vitreous and then to peel the internal limiting membrane off of the retina. When you do that, the ILM doesn't grow back. And in patients where that procedure is successful and the macular hole closes, the vision actually improves, and patients do not need to have that internal limiting membrane for the rest of their life. So in our work, we have developed a number of ways to remove the internal limiting membrane. It could be done mechanically, it could be done enzymatically, but we've shown that if you permeabilize or remove the internal limiting membrane, then when you transplant retinal ganglion cells, they actually do begin to send dendrites down into the retina and can form these synapses with afferent neurons that can then communicate visual information to them. So, we're thinking down the line of how we're going to translate this finding into humans. And one possibility may be that we want to either permeabilize or remove the internal limiting membrane as part of the stem cell transplantation procedure. That work will await further experiments in more advanced preclinical models.
It's fascinating, and it gets me thinking a little bit about what we've discussed on some previous shows about extracellular matrices and the differences between the ECM when we're younger and older, and the different proteins and so forth, and more the sort of the biophysical side of things here. And it's really exciting seeing you coming from the the biochemical, but also thinking about sort of all the biophysical components that go into this regenerative process for this complete functional integration. So it's really, really exciting, Tom. One other paper of yours that popped out, which I thought was really interesting, you published a couple of years ago in Current Opinions in Ophthalmology, 2019, and it was artificial intelligence in glaucoma. And once again, I mentioned my own experiences with sort of glaucoma testing ever since I was 9 years old. Obviously, that whole process now with these new tools, AI is hot everywhere in all industries, machine learning, computer vision, all this stuff. Where are we going in terms of some of these tools? I mean, this isn't the case where, you hook somebody up to this AI glaucoma machine here, you go have a coffee and come back and have the diagnoses and all that stuff. But where are we going with some of these tools in terms of supporting clinicians like yourself with these diagnoses and also with the ability to guide, where these appropriate treatments come into play?
I think it's going to play a huge role in the future, certainly of ophthalmology and hopefully of glaucoma. There's, again, a huge need here, going back to the fact that early diagnosis is so critical for glaucoma. If we can catch the disease early and lower the intraocular pressure, or maybe one day also give neuroprotective therapies, we can prevent vision loss and prevent people from needing things like optic nerve regeneration. But the key is finding it early, and we don't have great tests short of bringing people into the eye doctor's office to catch glaucoma early. So AI has the potential to change that as a really powerful diagnostic tool. And it already is doing that in ophthalmology in different diseases. So probably the best example for AI in medicine is for the screening for diabetic retinopathy. That's because you can take a picture of somebody's fundus, the back of their eyeball, look at the retina, and you're looking for things like little blood spots, hemorrhage, little spots where the retina is starved for oxygen, called an infarct, and other signs of the diabetic retinopathy process affecting the eye. It's very easy to take pictures. You can set up a fundus camera in a CVS or a Walgreens, and that's something that's being done. The tricky part so far has been finding qualified doctors to look at those images and interpret them. And that's where AI has really revolutionized things, because computers have been taught to look at those retina images and with a remarkably high degree of accuracy, judge whether or not there is diabetic retinopathy present or not. And in real time, tell the patient, you know what, there's a concern here, you need to go see an eye doctor, or everything looks pretty normal, you should probably have this checked again in a year. Ideally, we would have a comparable thing for glaucoma. The trouble is, glaucoma is a much more difficult thing to diagnose. It's not so black and white. There's a lot of shades of gray in looking at the diagnostics for glaucoma. So we've already talked about the fact that just measuring the intraocular pressure is not a good test because it can be low and you can have glaucoma. It can be high, you cannot have glaucoma. So the two main tests that we use as glaucoma specialists to guide staging and treatment of glaucoma are a test of the peripheral vision called an automated visual field test, or perimetry, and a scan of the optic nerve head, which measures the thickness of the retinal nerve fiber layer, which is actually composed of all those retinal ganglion cell axons that are going to be entering the optic nerve. There are other metrics as well in the retina. You can measure the thickness of the ganglion cell layer and the inner plexiform layer. And altogether, these provide us with information about the patient's structural and functional state with regards to glaucoma. Now, the problem is there is a fairly wide degree of anatomic heterogeneity in people. If I just looked at 100 people off the street, most of whom would not have glaucoma, they would have optic nerves that have all different sizes and shapes and different thicknesses of the retinal nerve fiber layer and different sizes of the cup, which is at the center of the optic nerve head. That's one of the measures that we use clinically to judge glaucomas, by looking at the size of the cup in relation to the neuroretinal rim of the optic nerve head. And as glaucoma gets worse, the cup enlarges because the neuroretinal rim becomes thinner. But people have all different sizes and shapes of cups and neuroretinal rims, such that there's a reasonable number of people that are followed by glaucoma specialists that we call glaucoma suspects. And what that means is they might have an optic nerve appearance that kind of looks like glaucoma. And we do an optic nerve scan, and they don't have a retinal nerve fiber layer that's quite as thick as the patients in a normative database that are bona fide to not have glaucoma and are age matched and sex matched. But it's not quite as severe as it would have to be for us to say this is definitely glaucoma. Mm-hmm. And so those group of patients provide a challenge for us because there's not really a good ground truth in order to train AI systems to detect glaucoma. When you have the situation with diabetic retinopathy, you can get 100 ophthalmologists to read a picture, and probably somewhere in the high 90s of those doctors will all agree that this is or is not diabetic retinopathy. If you have a bunch of glaucoma doctors look at a set of optic nerve photos and OCT scans of the nerve fiber layer and visual fields, there's going to be a surprising discrepancy in the early stage mild glaucoma patients as to whether or not they actually have glaucoma. And this has been done numerous times where teams of doctors have been shown the testing results for a array of glaucoma patients. And when they're definitely normal, there's very good agreement. When they have very severe glaucoma, there's very good agreement. But when they're in that early stage, the one that is most important to be able to identify for screening purposes, there's a surprising amount of disagreement and conflict as to whether somebody truly does or does not have glaucoma. What we do now is we follow those patients in clinic and we repeat the testing at intervals of maybe 6 months or 1 year. And if they stay the same for several years, then in retrospect, we conclude that they probably didn't have glaucoma. They just had an anatomy that resembled glaucoma. And if they actually show worsening of their nerve fiber layer thickness or their visual fields over years, then in retrospect, we could say, oh, they had glaucoma. And at some point in that monitoring period, when we recognize and determine that glaucoma is present, we'll initiate treatment for that. But at present, it takes consecutive visits and testing for a lot of patients to truly determine, do they have glaucoma? So, I think one way that AI might be useful is if it can detect things at a baseline time point that are invisible to the doctors. And there's good precedent for this. You may have heard when Google did some AI-based image analysis of retinas, they were able to tell things like whether that patient is a male or a female, which I can't do as an ophthalmologist. But something about the picture, and it's a little bit of a black box, was being recognized that they could tell the sex of the patient based on the retina photo. They could tell with some accuracy blood pressure of the patient and things that are totally not accessible to human minds that are looking at the picture. So one potential approach could be that you obtain photos at baseline and then follow those people for a few years so that we can divide them into the camp of these are the people that actually had glaucoma and got worse over time. These are the people that never had glaucoma because their testing was totally stable over years. And then maybe train the AI system on those baseline photos and see if they can actually differentiate something that the doctors can't see that tell you what camp that patient was in years prior. I think that kind of work remains to be done, but if that could be done, that would be really revolutionary for the field.
Absolutely. Really neat stuff. So, Tom, a couple days ago, I did an episode and we were talking about this this cute little amphibian called an axolotl, which has these ridiculous regenerative capabilities. You pull its eyeball out and it regrows an eyeball. You can scramble its brain up and the brain reforms. All sorts of wacky superhero capabilities that we as humans do not have. But it got me thinking of this book from a few decades ago called Shuffle Brain. And it was written by a professor at University of Indiana by the name of Paul Piche back in the 70s. He was actually in the School of Optometry. He was an anatomist by training. But his whole area of research was scrambling the brain of amphibians, basically, and trying to find out where memories and perception and all this other cool stuff resides. And, it got me thinking of your background. You are a neuroscientist. But you are also an ophthalmological clinician. At the beginning, you talked about sort of your awe at a younger age and understanding, where perception comes from, how vision happens and manifests itself. Once you solve all this glaucoma stuff down the road and you're focusing on other things, obviously the brain is a mystery, perception in many ways, consciousness, and so forth. These are all deep mysteries of neuroscience and ophthalmology. What other areas excite you in terms of some of these mysteries of not just the CNS, but of our perception, perceptual fields? What are some of those areas you'd love to touch on if you weren't spending time on on healing the world and in terms of glaucoma and all these other diseases?
Well, I'll tell you, intrinsic regenerative abilities, like you said, is something that's really amazing. It's not just amphibians that do this. Zebrafish, for instance, will regenerate their entire retinas. And so I'm interested in this now from the standpoint of what are the molecular pathways and what are the cues that allow some animals to do this, and why have those been lost? Why do mammals not possess that ability? And what can we learn from that might actually help our transplantation work? But I think in general, the emerging field of regeneration and aging and recapitulating youthful states is something that is really fascinating. DNA methylation and the methylation clock in cells, and how that relates to aging and potential maximal lifespan of human beings is really amazing. And the last 5 to 10 years have seen some really fantastic, interesting work done in that realm. And partially, I have been following that because it pertains to, again, optic nerve regeneration and vision restoration. There's some suggestion that resetting the methylation state of retinal ganglion cells can make them more resilient to injury and function better. But I think throughout the body, if we better understand the process of aging and rejuvenation, I think it really has the potential to provide treatments and unlock understanding of a number of diseases that would really help people widely.
Yeah, I completely agree on that front. It's a very unique time, and you're definitely at the epicenter of it all, at an awesome thought-leading institution, and it's really an amazing set of work you have going on. Before we wrap up, we've touched on. I tried to touch on many of the aspects of your research portfolio. Did I miss anything? And then also, are there any upcoming initiatives that we should know about in terms of conferences where we may run into you and talk to you, other talks you're going to be giving where we can learn more from you? Please, anything else that I missed, please take the floor, Tom.
Yeah, absolutely. So I think the one thing I wanted to mention goes back to our regeneration work and how, I mentioned that regenerating a retinal ganglion cell is a monumental task with all these parts that need to be put together. And the fact that there have been so many laboratories over the past 15 or 20 years studying discrete aspects of the regeneration process. So one thing I want to share with the listeners is a consortium that we've put together in the last 6 months called the RGC Repopulation, Stem Cell Transplantation, and Optic Nerve Regeneration Consortium, which is kind of a mouthful, but the abbreviation is RESTORE with 2 R's.
Perfect.
We have a website for the RESTORE Consortium. It's at hopkinsmedicine.org/Wilmer/RESTORE with 2 R's. And I have been really impressed with how this has come together. So the consortium is led by an organizing committee of 12 investigators from all over the world that have done a lot of the seminal work in stem cell biology, neuroscience, and optic nerve regeneration. And then there are over 200 participants right now in the consortium, about half of which are senior investigators that run their own labs and have been doing this kind of work for years or decades. And then half of which are emerging vision scientists who will be carrying out this work in years to come. We thought it was very important to get them on board early and foster mentoring and collaboration opportunities for them. So those are students, postdoctoral fellows, research associates, junior faculty, people like that. We've got 3 phases to the consortium, and phase 1 and 2 are actually complete. So in phase 1, from January to March of this year, we conducted an iterative online-based consensus-generating exercise where we identified the 5 most important questions or challenges that we need to tackle in the next 5 years to try to make retinal ganglion cell repopulation a reality. And there was 5 questions for each of 5 subtopic discussion groups. So we have one group on stem cell biology, one group on transplantation methods and models, one group on interactions with the host retina and neuroprotection, one group on interretinal wiring and synaptogenesis, and one group on optic nerve regeneration and brain connectivity. On April 30th, we actually had an in-person meeting in Denver just prior to the annual meeting of the Association for Research in Vision and Ophthalmology. And we all got together and discussed those 25 questions over the course of an entire day. It was an amazing set of discussions. People had fantastic ideas. I think overall we had more questions than we had answers, but that was exactly the point of the consortium, was to identify what we need to be doing and how we can be collaborating together to drive this work forward in the next couple of years. So we're currently working on writing up some white papers that describe what we discussed and what we're going to be working on in the next few years. And then the consortium is now in phase 3, which is going to include a number of virtual symposia and meetings, probably on the order of 3 times a year. And then we're hoping to, once again, have an in-person meeting in 2 to 3 years to see how far we've come during this time, where we've really tried to foster collaboration and really drive the field forward.
Outstanding. Outstanding. And we will, in the bio of the show, we will put a link to the Restore Consortium as well as to your bio, so people can find both that and you easily. Really, I mean, amazing stuff, Tom. I just, it's a fascinating portfolio, and really wish you the best with all of this. For everybody who's going to be listening to this episode across the various podcasts or watching on our YouTube channel, again, you've been listening to Dr. Thomas Johnson, glaucoma specialist, Alan and Shelley Holt, rising professor in ophthalmology, Wilmer Eye Institute, Johns Hopkins University. Tom, I want to thank you again for taking the time out of your schedule to come and walk us through the extensive portfolio you're involved with. Obviously, thank you for doing it and addressing this major unmet medical need. And as we say on our show, thanks for helping to create a better tomorrow for millions of people out there through your work. A really fascinating story.
Thanks for having me. It's been a real pleasure.
Really great to be here.